Today I would like to present you our ArrAI technology. It is the combination of an array-based high-throughput synthesis and screening technology and artificial intelligence. It allows us to map the chemical space and navigate through it to discover bioactive compounds as drug molecules. For people not very familiar with the concept of chemical space, it can roughly be defined as the collection of molecules whose synthesis is chemically possible. Even with the strictest definition in pharmaceutical sciences, a chemical space is a number larger than the total number of atoms in our universe, in the range of 10 to the power of 70. We will later call our AI model Ptolemy, named after the Greco-Roman astronomer, who developed the first qualitative geometrical models to predict the motions of stars and planets. This is the outline of my talk. I will first discuss the challenges and our mission, then introduce our team and the targeted market. After presenting our technology and a competitive analysis, I will present our business model and roadmap, then conclude with a summary and short and long-term goals. At the turn of the millennium, the 21st century had been promised to be the century of biotech. A quarter of century later, most of us might call it as the century of artificial intelligence. Although AI has shown its enormous strength in many fields, for example, AlphaGo, AlphaFold, ChatGPT, and so on, its capability in predicting chemical structures and functions, thus its utility in drug discovery, remain very limited. Every discovery needs a starting point, and in drug discovery we call it a lead compound. There are a lot of reasons making pharmaceutical data not ideal for building an AI model, starting from the early discovery. Screening campaigns remain costly and ineffective with limited scope of fixed-size compound libraries. There exists no technology that provides dynamically growing library with capacity to navigate to new lead compounds, the base for effective training with machine learning. Without an efficient and quick experimental feedback loop, most computational hypotheses will remain untested. An AI model cannot be evolved and trained by real-world dataset. We are building a platform which can synthesize and validate compounds on demand and in a high-throughput fashion. Thus, we can manage dynamically growing compound library, which is growing quickly and linearly with real-world data, and exponentially as a virtual dataset. The resulting AI model will be trained to help us to navigate the chemical space, to find bioactive molecules. Here I will explain why the feedback loops are essential for building a high-quality AI model, and why it is not yet possible for drug discovery. Motivated reinforcement learning is considered closest to human learning and creativity. We first have a dataset as input, a model is built computationally. The model will then generate new predictions, resulting in a new dataset. It is important to note that this is not only a new dataset, but also a new dataset according to the intermediate model. With the iterations of the cycle, our knowledge, or the quality of prediction, will grow exponentially. At certain point, mimicking will be replaced by creativity. We consider mimicking an interesting, but expected pattern, whereas creativity a surprising and unexpected pattern. Why this computational approach has little success in drug discovery? To discover a new drug for a particular disease, the scientists need to know the protein target associated with the disease, and to invent a chemical compound acting on the target. The chemical compound will be patented, and become the drug on the market. In the past century, advances in biology, chemistry, and physics have helped us to identify the protein targets, to produce the proteins, and to analyze their structures. 6 Nobel Prizes have been awarded to NMR and crystallography technologies to analyze the structures, while the most recent one on AI for predicting the structures when experimental techniques prove difficult. In contrast, much less progress was made on inventing chemical structures. Methods have been developed, allowing us to test many compounds, for example, high-throughput screening and DNA-encoded chemical library. However, different from proteins, which are globally stable and possess certain structures, drug-like chemical compounds are much smaller and dynamic. It makes both the human intelligence and AI equally difficult, if not impossible, to learn from the data. Let's come back to the loop. We have no problem with the initial dataset, using methods such as high-throughput screening or DNA-encoded chemical library, with library sizes from millions to billions. We can also build models using structure-activity relationship, molecular dynamics, or modern AI. Now we come to the bottleneck. We are very slow to make a new library according to the model and its prediction. It is commonly accepted that it costs more than $1,000, takes more than 1 day, to make a new compound and generate just a single new data point. ArrAI Tech breaks the bottleneck with a new automated synthesis and screening technology to make and test more than 10,000 compounds per day, less than 10 cents per compound. Thus, we can realize the automation of the iterative training cycles of protein-ligand cognition over many rounds and with large datasets. AI-assisted molecular evolution allows us to expand, navigate, and map chemical space with a dynamic dataset. We will also create a large portfolio of lead to preclinical drug candidates with high market value. Here I will bring you through the navigation process. It starts with the first library, for example, with 10,000s of compounds. After the assay and AI model training, it will make predictions in a larger chemical space. We will then choose 10,000 predictions with the highest scores as the second library and test them experimentally. The outcome will give us feedback to make the next training and prediction in an even larger chemical space. The iteration cycle will drive the exponential growth of our knowledge, thus the quality of AI model. Assisted by both computation and real-world experiments, we can map and navigate the chemical space to make discovery impossible for conventional technologies. In contrast to ArrAI, with other methods, the growth will be slowed down after the first round of input. Or to say, their searching of chemical space remains around the initial dataset. It is important to note that while ArrAI can work as a standalone technology, it also serves as a follow-up for the other methods, for example, with DEL or virtual screening, to validate the predictions coming out from them, as well as to further evolve the chemical structures. To realize the goal, we have assembled a team of chemists, AI engineers, clinical and business expert, as well as financial professionals. We are combining lab automation technique with AI to address the pharmaceutical market. In addition, we can also become a service provider for biopharma companies, which is the field of CRO, contracted research organization. Compared with the stable growth of pharmaceutical market, the CRO, AI, and lab automation markets are expected to experience a very quick growth in the coming years. The base, which allows us to realize the goals, is a technical breakthrough in generating and testing compounds on demand quickly and in a high-throughput fashion. It is a full package of technologies and know-how, including a patented material to make tens of thousands of droplets on surface as miniaturized chemical reactor, the in-house developed instrumentation, and software, as well as automation in imaging and analysis. It is a technology to synthesize compounds according to AI prediction, as array of miniaturized spots. The compounds can then be assayed using fluorescently labeled protein. Stronger binding will lead to stronger fluorescent signal. We can now make and test 2,000 to 5,000 of compounds per day, and expect to increase the throughput to 100,000 in 5 years. With this breakthrough, we can now test many predictions from the AI model. The 1 to 10,000 is still a linear improvement. However, once it is coupled to the iteration cycle, we can navigate through an exponentially growing chemical space and thinkable for any current methods. We are building a unique evolutionary and dynamic interface between experiments and computation. We have the patented array synthesis technology, in-house developed hardware, software, and extensive know-how. Our AI model Ptolemy is trained and evolved by the large and real-world screening data, and Ptolemy predictions are validated experimentally, also in a large scale. The iteration cycles of array and AI allow us to map a large chemical space and thinkable for wet lab and computer scientists. What makes ArrAI unique is not the large experimental dataset. It is the large experimental dataset co-evolved with the AI model. Therefore, the resulting AI model can map a chemical space and navigate through it unthinkable before. What also makes us unique is the low cost in testing and validating AI predictions. It gave us advantages in terms of cost, duration, and success rate, especially against those targets considered to be difficult for existing technologies. The technology has already been tested in many projects. Here I would like to use Claudin 18.2 as an example. It was published as a cover story in Adjuvant Chemie last year. Claudin 18.2 is a promising biomarker and therapeutic target for gastric cancer. It is a membrane protein without structural information, and none ligand was reported in literature. Therefore, it is a classical case as a very challenging target. We have performed for iteration cycles, covering a chemical space more than 10 to the power of 17, and got nanomolar binders. A compound was then tested in both gastric cancer cells and gastric cancer patient tissues, and specific binding and targeting effect have been validated. Drug discovery is a long, expensive, and rewarding journey, like finding a highly valuable needle in a large haystack. The business scope of ArrAI covers from the early stage discovery to preclinical study. By collaborating with external partners, some compounds will also reach the clinical stage. The first business direction, which shall be our focus in the first years, is R&D service. We will use our high-throughput synthesis and screening technology, ODAST, with AI-powered molecular evolution, to provide drug discovery service. Large collaborative projects with Big Pharma will be started in year 3. In addition to the income from service providing, the project will help us to grow our dataset for AI training. We will also run internal projects to build a portfolio of drug molecules. We will choose protein targets of high biomedical importance, with a focus on those very challenging for other technologies, as we have done with Claudin 18. We will select the most promising compounds to push through the various stages, from biochemical characterization, to cellular assay, to preclinical study, even to clinical stage in collaboration with pharma companies. The licensing fee, as well as the portfolio valuation, will increase as we move on to the later stages. We plan to run about 100 projects in the first 5 years, with more than 5 compounds reaching the preclinical stage. We will have a preliminary focus on oncology targets, also include the field of autoimmune disease and others. We will finish more than 300 projects by year 10, with about 20 compounds in the preclinical stage, and 4 compounds reaching the clinical phase. Regarding the form of drug, we will focus on peptide-drug conjugate, PDC. While antibody-drug conjugate represents one of the quickest growing fields in drug discovery, it has some disadvantages such as low stability, low oral availability, and high immunogenicity. By replacing ADC with PDC, we expect to circumvent these problems. It is important to note that different from most reported cyclic peptide binders, which are made mostly with 20 natural amino acids, our cyclic peptides have a broad choice of hundreds and thousands of building blocks. Therefore, it will give us a wide range of tunability regarding their pharmacological properties. The last business direction will be IT service. In essence, we are building an automated data generation system. The resulting dataset can be licensed to customers, for example, for AI, bioinformatics, and structural biology studies. We can also train and build ToLemi model for customers, for example, a ToLemodel for DNA-encoded chemical library users, or ToLemi display for phage display or mRNA display users. This is a roadmap for the first 5 years. In year 5, we aim to build a portfolio with many compounds in the preclinical phase. We will achieve the automation between the hardware and software, realizing an automated data generation system. In chemistry, we will be able to synthesize 500,000 compounds per day, not randomly, but according to the AI predictions. In AI, we will realize a unified model for prediction. In summary, we have an experimental method to test and validate AI-generated predictions in a high-throughput fashion. This is a technical breakthrough allowing for the iteration cycles to evolving bioactive molecules. The iteration cycle and resulting dynamically growing compound library make reinforcement learning possible for creating new chemical structures. The technology will be used to provide drug discovery service. Meanwhile, we will run internal and collaborative projects to establish a portfolio of drug candidates with high value. Through these projects, we will have an expanding dataset, as well as the Ptolemy algorithm. The dataset and algorithm can provide IT and cloud-based service. Most importantly, they will revolutionize the way how computation is used in drug discovery. Thank you for your attention. This is another angle of competitive analysis. At ArrAI, we can accelerate the validation process from less than one compound per day to thousands of compounds per day, and lower the cost from more than $1,000 to less than 10 cents per compound. The discovery process can be remarkably accelerated, as well as lowering the total cost. There are many other forms of chemical libraries for pharmaceutical research, from thousands of compounds to billions and trillions of compounds. However, it would be very expensive and cumbersome to expand them, not mentioning to expand them according to AI predictions. In contrast to the static libraries, with ArrAI, the fast validation process can provide enormous amount of information as feedback, resulting in dynamic libraries. It represents the most ideal form of library for AI-driven drug discovery. Here is a more detailed analysis to compare ArrAI with other technologies. Dell or other forms of display technology can provide libraries with ultra-high size. However, the readout of binding is indirect, and the binding can be affected by DNA. While both NMR and fragment-based drug discovery suffer from low throughput, high-throughput screening is associated with high screening cost. Virtual screenings, including various bioinformatics and AI methods, get their experimental dataset as input from public domain, which are highly heterogeneous. Most importantly, all these technologies lack throughput in validation. Thus, most predictions, either from human or from computer, will remain unexamined. Without the large datasets to power the training cycles, reinforcement AI training can also not be realized. It is important to note that while ArrAI has its advantage in high-throughput validation and enabling reinforcement training cycle, it can also synergize with other technologies. It can work as a standalone technology, but also serve as a follow-up for all the other methods, for example, with Dell or virtual screening, to validate the predictions coming out from them, as well as to further evolve chemical structures. Here is another view of market sizes. The pharma industrial has enjoyed a gradual growth for many years, and will continue as so. Meanwhile, we are at the age witnessing an exponential growth of AI industrial. AI sits at the intersection of them, and will become a powerful tool in transforming pharma and AI. Here is a summary of our ArrAI business model. We will provide drug discovery service for academia and biopharmas. It will also generate income from royalties and milestone payments with the partnered drug assets. Our dataset and AI engine will generate income with subscriptions and with tiered user plans. We will also provide service to train and build AI and machine learning models tailored for customers. Software as a service, as a cloud-based IT model, will generate recurring revenues. To build our own portfolio, we will bring promising drug candidates to preclinical and clinical stages to increase their market values. Late-stage development will be carried out with co-development partnership with biopharmas. Proprietary robotic drug discovery platform is also a commercializable asset. To describe ArrAI in a nutshell, we perform ultra-fast synthesis and screening experiments to validate drug candidates in a high-throughput fashion and to evolve drug molecules through iteration cycles. By establishing the feedback loops essential for reinforcement training, we can build AI-powered drug discovery engine. All these achievements are reflected by a quickly growing proprietary pipeline. We will generate income with drug discovery service and royalties from projects and partnered assets. the datasets and AI engine will bring income from subscriptions and licensing. The proprietary pipeline will create long-term value for the company. We will become profitable after year 5, and the valuation of our portfolio will be more than $200 million in 10 years. At the core of technology, we will build not only an automated synthesis and screening facility, but also a fully automated interface between the hardware and AI engine. So, the system will drive itself in testing and learning, and has the potential to become a new global drug discovery standard. We will have our headquarter and AI business in Zurich, Switzerland, and the wet lab and automated hardware facility in Dresden, Germany. Our co-founder Nathan Zhong is based in California, and the finance team is based in Shanghai, China. ArrAI combines German lab excellence with Swiss AI and pharma business hub, with a global vision of finance and innovation in its heart.